Okay, I think we are live. Wait a second. Let's wait a couple more minutes. I think we're still connecting. Some people are still connecting. Maybe a little bit. Mhm. Okay, I think we can start. I think people will connect as we go. In case if you have any questions, please ask them in Q&A panel, not in the chat, so we can address each question at the end of the seminar, at the end of the webinar. And the replay will be available online in a couple of weeks, and we will also send it for people who have participated or have registered. So the video will be available for a rewatch. Okay, so hello everyone. Thank you for joining our webinar on microfluidic automation for high-resolution microscopy techniques, featuring the DNA pain case study. We are here joined by Philipp from ZEISS Research Microscopy Solutions for life science applications. Thank you very much, Philipp, for your expertise and attending this webinar. In this brief webinar, we'll introduce the key advantages of fluid delivery and control in microscopy workflows and explore different techniques that can be used with the fluid delivery and microscopy. We'll then take a closer look at Fluigent's Aria, how our automated perfusion platform designed specifically for microscopy, highlighting its technical characteristics and use, benefits of its use in complex imaging protocols. And after that, Philipp will walk you through the ZEN software, how it communicates with Aria, and its different range of functionality that can be applied, for instance, in small molecule localization microscopy, also known as DNA case study. And we'll finally wrap up with a short summary and open floor for a Q&A session to address all the questions that you might have during the webinar. So let's start from basics. Most staining protocols are highly repetitive. Here it's a given example of immunostaining. A classic immunostaining workflow typically involves a long sequence of manual steps, starting from sample preparation to the fixation with paraphalmadehyde. After, you might need to permalyze your cells for reagents to enter the targets, like then blocking with the BSA or serum to avoid the background staining. Next, you'll do the incubation with the primary antibodies, sometimes overnight with the secondary antibodies, etc. And all of this also include the concentration optimizations for sample types, the washing between each step with PBS, with Twin 20. Finally, you do a mounting, a contrastaining with DAPI, etc. And all of this, throughout all of these steps, the user is pipetting everything manually, carefully trying not to dislocate the sample, not to dry it out between the steps, to obtain this type of beautiful and informative images. It can be time-consuming and tedious, and at the heart of all of it is a manual pipetting, which comes with several challenges. And first, I'd like to introduce the pipetting risks and errors that can come with the manual pipetting. It's not only tedious, but also it comes with some of the risks, like touching the slides if you're working with the live cells, and sample contamination, under delivery of reagents, depending on if the tip has a low wettability. So the physical aspects of pipette tip pipetting at an angle also can cause inconsistent volumes, and can even tip wiping can cause loss of sample, which is very valuable in this type of protocols. And beyond it, we have also the pipetting variability, which is is known that even experienced users have 5.7% intra-operator variability and 8.1% inter between users, variability in between users. And when it comes to integration with the microscopy or manual pipetting that has to be done on stage, it also comes with the danger of moving the microscope blade, especially shifting the field of view in case if you're acquiring stacks or having a multiplexed experiments. A major problem also could be on stage with live cell experiment, the liquid spills, and sample damage during the handling. And the liquid spills can be very problematic when you're working with expensive objectives, etc. So all of these points point us towards the need of automated and precise liquid delivery system, fluid delivery system, especially involving high resolution microscopy, multiplexed imaging, live cell imaging, etc. And microfluidics here comes in handy. These platforms are among the most precise and programmable solutions for fluid delivery. They offer exceptional control for microfluidics and imaging workflow. They're programmable. However, they can come with some requirements. For instance, if we look here, it's building and maintaining this type of platforms, setups comes at cost. And it's not just funding, it's not only financial, but also it is in time, expertise, labor, and it requires training the personnel, troubleshooting experiments, and setup optimization. And life science experiments are already difficult as they are. They're complex. Therefore, we do not want to add this type of complexity to the setup. And therefore, we have Aria. Fluigent developed Aria to overcome the limitation of manual pipetting and complex setups in multiplexed tissue imaging. And it provides a platform. Here you can see a platform with fluids delivered to the chip. Aria integrates both hardware and software, allowing users to connect directly to the imaging chips and automate the multiplex tissue staining. Beyond that, it can be configured and communicated with the microscope, enabling like a workflow synchronization that is essential for live process samples, freshly conjugated markers, and multiplexed protocols, which we'll talk in detail later. By combining this programmable fluidics synchronized with imaging, you can simplify the protocol and have a high throughput multiplex tissue staining and imaging. So Aria is designed to speak the language of biologists, making it easy to turn a traditional notebook protocol into automated digital workflow. You can see here, for example, here you have inject paraformaldehyde, 100 microliters, 200 microliters, and next step is a wait step to incubate for self-fixation. Then you'd add the next step, which would be the volume injection of your antibody. So it can precisely deliver the liquids and transfer this type of protocols into a software which can run automated on its own, hands-off. And one of the protocols supported by Aria is IBEX, an iterative immunolabeling method that enables multiplexed imaging over 65 markers across diverse tissue types. On this slide, you can see the human lymph node. This protocol was published in Nature Protocols in 2021, and Aria played a key role in automating this fluid delivery in sample preparation. According to the testimonial of the researchers, the system increased by 2.5 times the output of the number of samples processed during a week compared to manual methods, which significantly speeded up their workflow. We also have a webinar with Colin Chu, the researcher who is a co-author of this paper. We delve into the technical details of multiplexed imaging technique, and I encourage you to watch this webinar on our website. It also will be shared to you as a link after the webinar. So you can look in the detail about the IBEX protocol. Moving forward, it's not only IBEX protocol that requires the iterative workflow. There are more cyclical imaging methods. So it can benefit from automation. Like here, you would label with a particular marker or antibody, acquire an image, and bleach or remove your sample and repeat the cycle again, which allows you to have multiplexed tagging with a marker of an interest. Some examples are cyclic immunofluorescence, Codex, 4i, exchange paint, and SecFish. We look at multiplexed immunolabeling technologies, and multiplexed labeling technologies, they might seem very different on the surface, whether they're working with different type of labels, a DNA barcoded antibodies, imager strands, reporters, barcodes, fluorophore conjugated secondary antibodies. But we always have a marker, and this workflows also vary a bit with the imaging modality, whether it's a confocal microscopy or epifluorescence, spinning disk, super resolution microscopies. It can be supported here as well. And finally, you have a step of inactivating agents. It can be also different in chemistry. It can be an optical bleaching with a laser. It can be a chemical bleaching. Depending on the type of label and the conjugation, you would have a different removal method as well. But overall, the important part is that no matter which technique you use, whether it is SecFish or Codex, or what imaging modality you would choose, perfusion is an essential step to control and automate the delivery of the reagent and the removal of the reagents, so you can trust the results, preserve the probes that you're using, and have a high precision in your reagent delivery. And Aria can seamlessly integrate into this workflow with gentle sample handling, especially when we work with more sensitive reagents such as DNA or RNA. Now I'd like to pass the floor to Philipp from ZEISS Research Microscopy Solutions to share his expertise on how to communicate Zen microscopy with Aria, but also give us an interesting overview of DNA paint use case.
Yes, thank you very much, Anel, and thank you for the opportunity to talk here also, and thank you for the audience for listening. And yeah, let me first share the presentation. Not very. So, can you see the presentation? Anel, did this work? Yeah, you can see the presentation. Okay, you can hear me. Great, let's jump right into it. As Anel already said, we can look at two things. First of all, the integration of the Aria software with the imaging control software from ZEISS for the most light microscopy platforms, ZEN. And then let's look at the DNA paint application example that we created. So yeah, I hope Anel already convinced you that combining microfluidics and imaging can lead to great applications and can produce great value. That was also the motivation on ZEISS side some years ago to look at the market and see whether there is providers of microfluidics that like to partner up more closely and develop this relationship between imaging and microfluidics more closely. We're very intrigued by the offering from Fluigent. So both the Aria hardware, the Aria instrument, for its compactness, for its all-in-one approach, for its general usability, and don't need to be an expert to use it. We really like this. We also like the workflow software. Thought it's really nicely done and very intuitive and speaks the same language as your lab book protocol would. We were not so happy in the way that, the Aria software was speaking to ZEN in order to create combined microfluidic and imaging workflows. It could already speak to ZEN, but only via so-called TTL signaling, which has some advantages, but in most cases has disadvantages. First of all, you require, at least on ZEISS side, you require additional hardware. You require a trigger box. And also the way the user sets up the workflow is now kind of broken. It doesn't do it in this nice approach anymore with this workflow scheduler. He has to switch into ZEN and basically create a second workflow for the imaging and configure all the synchronization between the workflows manually. Yeah. Also, the TTL signaling, you needed to know what you are doing. You needed to configure the TTL signals themselves, and if you yourself, and if you didn't do that right, then the signaling was quite error prone. We thought we could do better together, based on the idea that, both Aria software and ZEN software typically run on the same workstation when they run next to a microscope, if the Aria is next to a microscope, and that both of them have other communication protocols available. We started developing some special ZEISS features here. So first of all, a real acquisition block to add to the workflow, which makes the readability and the setup easier. And then this acquisition workflow automatically detects and reports here in a drop-down list, or lets the user select the available experimental setups from ZEN. So ZEN is running in parallel. Here in the drop-down list, you will have all the experiments available in ZEN. So the interaction logic is then quite nice. You build up your workflow in the Aria software. You configure the details of your imaging experiment still in ZEN, but that has advantages. People can do as they are used to in the acquisition setup in ZEN, and they can do any experiments of any kind of complexity that is covered by ZEN and the instrument itself. Yeah? So it doesn't matter whether this is a time series, C-stack, or whatever kind of complication of an imaging experiment, it will be compatible with this workflow. Because the idea then at runtime is that the Aria software controls the microfluidic flow, and in due time, it will then send a command via TCP/IP to ZEN to initiate an experiment, and also, synchronize then the further workflow, further microfluidic workflow. Yeah. So that's really nice interaction logic we find, and much simpler than TCP/IP, TTL. If people, for whatever reason, still need TTL, maybe because they need very fast responsiveness of the instruments, they can still use it. But if they want to go more usable and easy to set up, they can use this. Yeah. So, and also the setup of TCP/IP communication protocol is very simple in both softwares. As I said, they are equipped with this communication feature, and you just have to go to the option enable TCP signaling and make sure it's the same port that they talk to each other. Everything else happens in the back end. So, and that leads to, yeah, a very nice workflow setup, very nice usability. And I like to show this just with a little experiment here. The experiment doesn't have a lot of meaning. That's just some cell culture. It's two live stains that are supplied one after the other and acquisitions in between. Yeah? So focus on the ease of setup, let's say like this. So where do I start this? Does it start? I make it started.
I think it's on the bottom. Yeah,
Yeah, there you go.
the
Yeah.
button.
So first you configure your experiment as you need in ZEN with all the features. Then you would go over to the Aria software. And in the Aria software, first of all, you configure your microfluidic setup. As I said, this is just a two-step staining protocol. You have MitoTracker red, washing, after that an acquisition. And in the acquisition, we can directly select then the experiment that we just created. And you see you have this workflow done in the matter of minutes. Yeah, so it's really easy to set up and intuitive. So, and then, yeah, you press run and you have a walk away solution. You don't need to sit next to the instrument. Wait a sec, it's wrong again. This is the one. Yeah. So let's go. The Aria software from that point on controls everything you need. Yeah. It controls the fluid delivery. It initiates the acquisitions. It does another fluid delivery. It does another acquisition. And yeah, you enjoyed your coffee while that all happened. And you can have automated workflows if you want for days on end. So as a little side note, really not going into depth here, but there is actually two options for communicating between the machines or for executing imaging within the Aria software. The first I just explained, that is the standard version, very easy, also for non-experts. If you want to do a little bit more with your imaging, let's say you have some custom steps that you want to have before the image acquisition or after that. Here's an example I wrote down, and this is a real use case. We did that actually with some other customers, a custom autofocus, and then some post-processing afterwards. If you want to do something like this and you have some level of expertise, you know how to write ZEN OAD macros, you can do that. And then from the Aria software, you directly call a macro now. So that is the second interaction logic that can take place. So, and yeah, the nice value proposition coming out of that is that more or less out of the box, Aria and a large number of our optical systems in light microscopy are compatible with each other, yeah? Because the communication between the softwares really just relies on this open application development, TCP/IP communication that ZenBlue always has, and then on the instrument-specific acquisition setups that ZEN will always be able to run. Yeah, so you can select your optical system based on your application. So, and then let's jump into one of those applications, DNA Paint. And this application note or this application story we did together with experts of single molecule localization microscopy. That's the umbrella term. The Ralf Jungmann lab at the Max Planck Institute for Biochemistry in Martinsried at Munich. And special thanks then for the people that were willing to work with us, which was colleagues from Ralf Jungmann from his lab, Kimberly Kramer, Heinrich Grabmeier, and Isabel Bautrexel. Yeah, I like to also, for people who never heard of DNA Paint, give the full picture of the story. So go at least into every crucial step. So let's start with some theory. So what is single localization microscopy? You know that every fluorophore is producing an image that is spread out, that has a certain point spread function, which is normally limiting the resolution, which is normally a size of some 200 or 300 nanometers. But as long as this point spread function of a single fluorophore is isolated and does not overlap with some other point spread function, still you can calculate with high precision, with sub-pixel precision, the localization of that fluorophore. And that is the idea of single molecule localization microscopy. In a real sample, however, you will have fluorophores typically overlapping. So somehow you have to find a way of having those fluorophore switch between active and inactive state, or also called blinking. Yeah? And make sure that with every single acquisition, you only have a subset of fluorophores active, and you will have isolated PSFs. Then you do a lot of acquisitions, you do a lot of frames to collect all the random blinking from all the molecules, and you will be able to have a super resolved image in the end. In DNA Paint, so you can do the blinking with different chemicals. In DNA Paint, you use the features of oligonucleotides that, based on complementary sequence, can bind each other, but do so reversibly. So they bind, they go off, they bind, they go off with a certain kinetic. And that's perfect for single molecule localization microscopy. The idea is now every target molecule is decorated with a so-called docking strand. Then you add imager strands to the solution, and only a subset of imager strands at any given time will reversibly bind the docking strand, will become detectable by the imaging. If the molecules are in solution because of Brownian motion, you will not be able to detect them. So here you have a natural blinking in DNA Paint. So DNA Paint also has its version, which is multiplexing, which is actually a cyclic multiplexing. The idea here is, the intention is to detect several targets in the same sample. And you can do that by first treating the sample with all kind of docking strands for all target molecules, and then supply sequentially the single imagers. Yeah? First, I supply the imager for the first target. I do my multi-frame imaging experiment. I add the imager for the second target. I do my multi-frame imaging experiment, and so on. I arrive at a data set which is multidimensional, which is multiplexing. And as you can see here, you have a cyclic process where microfluidics really can help you. And exactly that is what we did in this experiment. So we had an Aria 7 available as our super resolution machine. We used a chip from IBD. That was not a special chip in any respect. The only thing that we made sure was the optical setting, or was perfect for the optical settings, meaning that it has a glass bottom with 170 micrometers. Other than that, any other chip could be used. Chip is connected then to the Aria, and the Aria does the microfluidics. This would be the workflow setup from the perspective of the software. So the Aria software would control in a cyclic process the liquid delivery of the imager, then initiate the image acquisition, and once that is done, do the washing, and then the next step starts. We can have a look really fast on how such a workflow would look like, and the key features of the workflow from the perspective of the Aria software. So here you could see in a cyclic protocol, you could really use loops. Yeah. So loops let you allow to run a certain sequence multiple times. And so here we use the loop running for 4 times because we had 4 images. In every loop, obviously, we want the acquisition to take place. We in every step here, step number 1, or in every cycle, we want washing to take place. Also, we do some additional flush tubing to clean the machine. But then also the scheduler allows to set variants within the loops, and this is where the specificity in every cycle comes in. Yeah, so here we define which imager is supplied in every cycle, which are here called R1 to R4. Yeah? So that would be the protocol. Relatively easy to set up, relatively easy to read. And then, yeah, let's just go shortly also in the image reconstruction, because actually, so if you have single molecule localization microscopy, your raw images or your raw time series looks something like this. It's just like Christmas, so a lot of blinking. You see that certain positions here are not blinking. And this is not target molecules. These are so-called fiducials. They are obviously always active. And they are supplied to the sample in order to support the image reconstruction. We will see in a step why or how. So the first step in the image reconstruction, which by the way was done in this case by with the help of a freely available Picasso software, also coming from Jungmann Lab. So here's the GitHub page for that. First step in this reconstruction would be from the raw image frames, from the single blinking events, do the localization, as I explained, as the key step of single molecule localization microscopy, get this sub-pixel localization of all the blinking events in the data set. Now, if you look at a maximum projection of those single localizations, you will find that you, don't see much. You only see some patterns here. And the reason is we are here in a high resolution, small field of view, high objective situation where just a little bit of drift can change the field of view. Yeah, and this is exactly what happened here, and that is why we use the fiducials. We have now here the pattern of the fiducials over the whole period of the image acquisition, and we can now normalize this in a second step. So drift correction, and then we have drift corrected localizations for the fiducials, obviously, but more importantly, also for our blinking events or for our target molecules. Now they become visible. Here they haven't become visible because they were drifting and therefore not giving one strong signal. So, and that were the key steps of the reconstruction. Then I can show just 2 results. So we did first of all an experiment with DNA origami, which is it's a synthetic structure, synthetic self-assembling structure made out of single oligonucleotides, and it's a tool that is used in this case because of its nice spatial features. So this is a grid-like structure with very defined distances between certain points. Yeah, so the docking points for different images are 5 nanometers apart, and the docking points of the same images are 20 nanometers apart. So you can use that to validate the quality of your super resolution. And for us, it was a validation that we were successfully able to run a 4-cycle sequence with the Aria, detect all the targets as we wanted to, and have a quality that is also comparable to what you normally would expect from this method. So a localization precision of roughly 5 nanometers and a resolution of roughly 10 nanometers. You can do the same thing, and that's probably more important, also with cells or with biology sample. In this case, it's the U2RS cells, and 2 targets were detected. One of them mitochondria, TOM20, the other one nuclear pores, NUP96. And you can see that the cell with exchange paint has been stained in both channels or both dimensions. And you can also see from the inlay here that we actually have quite a nice super resolution quality. We can see the single components, the single subunits of the nuclear pores. If you quantify it, you will end up also with something like 5 nanometer of localization precision and 10 nanometer of resolution. That takes me to some take-home messages, for at least for my part. So we really value the Fluigent Aria because of its all-in-one, flexible, affordable, and plug-and-play approach. It's intuitive instrument operation without no microfluidic expertise required, and its optimized integration with ZEN-based ZEISS instruments. And there you can then benefit from the optical quality of our instruments. For the DNA paint application, the conclusion is Aria is well suited to support DNA paint, especially cyclic protocols like exchange paint, or if you want another one to get inspired, resolution enhancement by sequential imaging, RESI would be another recent method that also runs cyclically. Workflows with Aria achieve similar localization precisions and resolutions as manual operation, but with more automation and robustness. So we just recently started to really market or advertise this Fluigent ZEISS solution. So we're just in the beginning. But what I can tell you, if you're interested in the solution, ZEISS Customer Center in Oberkochen is already equipped with one. So you and they are actively developing suitable demos. So you might get in contact with them if you want to see more. Or if you're really interested in purchasing a microfluidic system together with ZEISS instruments, please contact your local account managers or product manager, Deborah or me. And yeah, we have more marketing material and more product information available, sales presentation, quick start guide, further collaterals, which will be launched soon. But upon request, it's available already. And then obviously you can have much more information from the Fluigent webpage. And thereby I say thank you and give back to Anel.
Thank you, Philipp. It was very fruitful, the presentation of the DNA paint. You can see my screen. So to finish up, I'll give you a technical specification of Aria that might answer some of your questions about the use and introduce Fluigent shortly. Aria is a fully integrated solution that includes the valve that you can see here, the flow unit, the flow sensor, and all of the tubings, and the rapid stop valve that is integrated here as well. You can see all of the components that can be done and assembled in an Aria in a box, let's say. We offer 2 variants of Aria for a single output version, specifically for multiplexed imaging, where you can have more than 10 fluids perfused in a single chip or in a single flow cell. And the serial output, which allows perfusion of multiple samples, not at the same time, but one by one, which helps to maximize the number of samples or sample preparation for other type of applications with, again, 10 samples to perfuse and 10 reagents to perfuse and 9 samples at the same time with a switch valve. So what makes Aria especially great for microscopy application? It is precision and accuracy on the flow control. So it has a 5% of measured value variation at flows below 40 microliters per minute, and reproducibility of plus minus 0.5% of measured value. So it's very reproducible fluid delivery. And here you can see, as I mentioned, we have internal on-off valve that prevents the backflow during the incubation time. So when the pressure and the flow drops, it's almost instantaneous. So we'll have a little no backflow, which is a plus, is an advantage of a smart delivery system, which means the internal volume varies depending on the area that you're using. It varies by 10 to 20 microliters of an internal volume. The total amount can be between 100 and 150 microliters until the chip inlet. So all of this is automatically calculated by Aria as a calibration when you turn on the instrument. And when you run your experiment, if you work with, for example, 150 microliters of antibody that you perfuse in your flow cell or microfluidic chip, the solution will reach the chip at exactly specified time and incubate it for exactly specified time in the workflow. I find it a very useful feature because you don't have to calculate all of the tubing internal volumes. It is done automatically on the software, and you only care about the protocol that you're designing. And Aria does the rest. So talking about the protocols, you can design, as Philip mentioned, loops, which can be very helpful for multiplex imaging experiments with variation, which type of reservoir you want to perfuse at which time. So here, for example, 7 iterations with a variation of first and second iteration to use reservoir 6, which could be with your marker A, and then the rest you want to mark with a B in iterative manner. We have timed injections that allow you to choose specific flow rate for a specific time. The volume injection, I think, which is most commonly used to inject 200 microliters and to incubate with a weight function, for instance. The group exists for visually grouping your experiment and allowing you a more user-friendly interface. Aria also can send the TTL signals for external devices, like a microscope or other instruments that you can control and be a master point to control the microscopy or, for example, a temperature control, etc. Overall, these features give you flexibility to automate your complex experiments and to iterate specific perfusion steps. An overview of Aria, it gives you different dense solutions. We can have a standard or custom protocol. It allows a very high precision and very high reproducibility for the fluid perfusion. It saves the time. All of the fluid delivery is automated, and it's user-friendly, it's intuitive use, it can be communicated with ZEISS microscopy, with ZEN, across different microscopes, and with the TCP signaling, it can be even more user-friendly compared to TTL. So that's the advantages overall of use of Aria. You can learn more on our website and email us if you have more questions, and we'll answer also Q&A session in the panel. And finally, to end the webinar, I want to quickly introduce our company, Fluigent. For some of you who might not know, we are in the industry of fluid control at the microscale level, when it's crucial in life sciences. And not only in life sciences, we empower thousands of flow control. We provide thousands of flow controllers worldwide. We have presence in US. We have an office in US subsidiary in several distributors all over the world. Our products have been widely used in top research institutions. We have over 3,000 scientific papers featuring our products. And we keep developing in mind with having microscale fluid control in mind. I think I can give a small poll just to see if you used a Fluigent technology before. You should be able to see the poll right now, and I encourage you to answer. Okay, 100% answered. I know. Still wait a little. I will end the poll. Surprisingly, around 60% of people are not familiar or have not used Fluigent technology yet. So it is a great opportunity for you to learn about our products. And now we'll pass to Q&A. Here you can see the contact@fluigent.com email and my personal email. Please reach out if you have any questions. Okay, so thank you very much for your time. We are available at our emails that were mentioned, the contact@Fluigent or our personal emails. Please feel free to reach out with your questions, and we're happy to learn about your applications as well. Thank you very much, Philipp.
Yes, see you.
See you. Bye. Thank you.